your mobile devices on silent for the duration of the conference. Thank you very much and welcome. Dr. Juan Mosqueda is a veterinary certification graduated from the Autonomous University of Querétaro. He holds a Doctor of Philosophy degree from the doctoral program in veterinary science at the College of Veterinary Medicine, Washington State University. Currently, he is a full-time professor at the Faculty of Natural Sciences of the lab. He is responsible for the immunology and vaccines laboratory of the same university. President of the Association of Veterinary Parasitologists, AC 2014 and 2017, appointed expert in the reference laboratories of the World Organization for Animal Health in Mexico of Senacica for bovine babesiosis and bovine anaplasmosis from 2014 to the date. Member of the Mexican Academy of Science, member of the Mexican Veterinary Academy, coordinator of the Veterinary Immunology chapter of the Mexican Society of Immunology, member of the National System of Researchers Level 2, leader of the Consolidated Academic Body, Animal Health and Environmental Microbiology. Give a round of applause.
Good afternoon. Thank you very much for the invitation, especially to Vinada and the organizing committee. I would like to start my presentation telling you that I want to divide this talk talking about an introduction of vaccines, what are vaccines, what they are used for, and then some basic concepts of immunology, which most of you may know, some others may not. And then I will proceed to tell you about an example of how we develop vaccines in our laboratory. Yeah. What is a vaccine? A vaccine is defined as a preparation of many things, like organisms. They may be attenuated, alive, or inactivated in case of viruses, or their components, proteins, amino acids, carbohydrates, nucleic acids recently. And when they are administered, they stimulate an immune response. But the goal of a vaccine is that a vaccine has to be able to protect an individual in the future, whether against a pathogen or a substance like a toxin, but it has to be able to confer protection. Are vaccines important? Well, you know that there are recent arguments whether vaccines are not useful or they are dangerous, but it may be that people don't exactly know what is the importance of vaccines, but vaccines have had a huge impact on human and animal health. For example, imagine that clean water is considered the most important basic human right. But just behind clean water, vaccines has had the most impact on human health. And that tells you the importance of the use of vaccination. This table examples the success of vaccines. This is a table that records the cases of the most important diseases in humans in the US, the cases per year, and you see the pre-vaccine number of cases, the post-vaccination once the vaccines were developed, and the percentage of reduction. As you can see here, for some diseases like smallpox or diphtheria or polio, the percentage reduction is 100, so no cases appear after a vaccine was developed. For diseases that are even more serious, like measles, for example, which in the US they have almost over 500,000 cases, they had a reduction of 99.98%. That means that we really don't conceptualize the importance of vaccines or the importance that vaccines has in our society. 200 years ago, for example, the life expectancy of a human was only 37 years. And besides clean water and vaccination, we have had increased our life expectancy for several decades. What is an ideal vaccine? We want to produce vaccines that are safe and that have many other good attributes. But what are these attributes? An ideal vaccine has to be 100% safe, which means that if you immunize somebody, this individual should not present any reaction to it. A single dose should be sufficient, but most of the vaccines currently, they require 2 or more doses. They have to be easy administration, you know. If you have a syringe and you have a needle, that is really complicated, especially for vaccination, for vaccinating animals. It would have an indefinite storage at room temperature. Most of the vaccines, they have at least, they require a refrigerator. They have to be easy and cheap to produce, and they would have to be able to confer long-lasting immunity. Ideally, the total length of the life of an individual. For example, in humans, it would have to confer protection for 70 or 80 years. Well, there is no vaccine in the world produced for animals or humans that are 100% ideal. So that means that we need to get higher quality of vaccines nowadays. What is the time for that it takes us to develop a vaccine? People, they don't realize that developing vaccine takes many, many years. For example, here you can see that for varicella, it's 28 years. The vaccine for flu is 28 years. Papillomavirus vaccine is 15 years, and rotavirus is 15 years. Only for COVID, we had 18 months, but just remember that for COVID, it has already been studied for 20 years because of the previous viruses that appeared 20 years ago. So they knew how to do vaccines for coronaviruses for many years now. So it really takes a long time to produce a to develop a vaccine. But I would like to also show you these basic concepts. What is an epitope? Does anybody can raise their hands if you know what an epitope is? You know what an epitope is? Good. All right. So there is a definition here. An epitope is defined as a part of an antigen that binds to a specific receptor and on B cells or T cells or on secreted antibodies. This is an antigen, it's a regular antigen. One would expect that the immune system would detect the whole protein, but the immune system doesn't detect the protein. It does only detect small parts of the antigen called epitopes, which are these ones in a gray color here. And if you can see the antibody, this is an antibody, it can recognize these linear epitopes over here, like this, or it can recognize conformational epitopes like that. So epitopes are very important because they are the only parts of the antigen, they are the only parts of the pathogen that bind to antibodies or to T cells. There are very few epitopes in an antigen. Like you can see here, this is the spike protein. You can see the epitopes in this protein. These are the only ones that are bound by T cells or by B cells. But each epitope has a different immunogenicity, which means that each epitope produces or induces the production of different levels of antibodies. That means that only dominant epitopes generate antibodies. The subdominant epitopes are less immunogenic and they don't generate an immune response. And this capacity allows the pathogens to produce hypervariable immunodominant epitopes that are not recognized by the immune system over time. So in my laboratory at the university, we have had many years of experience identifying epitopes from different antigens, from different pathogens that cause disease in different animals. So we use a sort of a bioinformatics approach and also immunologic approach to identify B cells or B cells in different antigens. We then identify these epitopes. Remember that the epitopes are small parts of a protein. So this is an epitope, this is another epitope, this is another epitope. And we then use the coding sequences of those epitopes and we assemble them in a linear sequence. And then 2 or 3 peptides or 2 or 3 epitopes from each antigen are included, which means that these 3 may be from one antigen, these other 3 may be from another antigen, and so on. And then 1 to 7 antigens are included, and we develop then a synthetic gene, which we design, we patent those synthetic genes, and then they are used for expression as recombinant proteins. This recombinant protein is what we call chimera. Remember that the chimera was a monster from the ancient Greek mythology that was a monster made of a head of a lion or a head of a goat with a tail of a dragon. And this chimera is more powerful than the lion or the goat or the dragon alone. So we call them chimeras because these vaccines are more powerful than the vaccines made of one single antigen. How they work? The chimera is inoculated or is used to vaccinate an animal like this, and then the immune system of that animal or that individual develops antibodies against every single of the parts of the chimera or to every single of those epitopes. So we have a protein that induces an immune response against many parts of the pathogen at the same time. So there was kind of an introduction of what I wanted to talk to you about, but also I would like to move to tell you how do we develop vaccines in our laboratory. I choose a disease that is very important in Mexico and in many parts of the world that is called bovine babesiosis. Have you heard about bovine babesiosis? Bovine babesiosis is a disease that impacts livestock industry worldwide. It causes huge losses because these animals get babesiosis, they stop eating, they stop drinking, and then after a couple of days they die. It's caused by intraerythrocytic protozoan parasites of the genus Babesia. These parasites are transmitted by ticks, and the disease is characterized by fever, hemolytic anemia, hemoglobinuria, and death. At the end of the 18th century, Victor Babes, a medical doctor, was called by the Romanian government to attend cases of cattle dying in Romania. And what he found, looking at blood of infected cattle was small dots on the blood of the cattle that he called Hematococcus bovis because he thought it was a bacteria. But Starkovici, a collaborator of Victor Baves, renamed this protozoa as Babesia bovis. And then two researchers in the US, Smith and Kilburn, discovered a second species that they called Pyrosoma bigeminum that later was renamed as Babesia bigeminum. These are the two most important species in America, but in Texas, they even wrote this article talking about the Texas cattle fever or the southern cattle fever, which is how they call the disease in the US. If you see a smear of a blood of infected cattle, you will find some protozoa inside of the erythrocytes. These are erythrocytes of cattle that we grow in vitro. I took this, I cultured these parasites. We maintained the parasites culture in vitro by feeding them with erythrocytes from healthy cattle. This is Babesia bigemina, this is Babesia bovis, and these two are the most important species in the world because they are the ones that have a wider distribution, and they are the ones that are in Mexico. But there are other species like this long list. We have Babesia divergens that also affects humans. We have Babesia ovata. This is a species that is very common in Asia, was discovered by a Japanese researcher. And this species is maintained in a laboratory in university in Japan. These are the vectors. The vectors are ticks, Rhipicephalus ticks. This is a female, 21 days of age. When they are very, very tiny, they adhere to the cattle, they start feeding, and in 21 days, they grow this big, the size of a nail. And this is a male, an adult male, same age. Just before dropping, they mate. The male goes and looks for another female, and the female drops to the ground, and then it lays up to 5,000 eggs and dies. But Babesia goes into the embryos, and then when the larvae hatches, the larvae is already infected. The larvae goes into the grass and then waits for a cattle to pass. When the cattle is grazing, the larvae attach to the cattle and transmit babesiosis to the cattle. What is the importance of this disease? It's really huge, but I will just show you one example in Mexico. In the world, we treat babesiosis by killing the ticks. How do you kill the ticks? Using acaricides. How much money you spend on acaricides? The global consumption of acaricides in 2003 was $70 million US. Of these $70 million US, Mexico invested 17 million used to buy acaricides to treat the cattle to avoid them from getting babesiosis. But that is not the problem. When a farmer in Veracruz, where I took this video, buys a male, a bull from Canada, 100,000 pesos, and he brings it to Veracruz, he dies in 10 days of babesiosis. And then the farmer, he doesn't want to buy another cattle anymore. So he prefers to have this cattle that they don't produce any meat or any milk. So the real impact of this disease is that it prevents introduction of high quality, genetically high cattle, which are very susceptible, and it delays the development of the livestock in Mexico. Let me see if I, no. Can you play the video? Like that. You see, these persons are treating this cattle with a parasite. They don't use any gloves. They don't use any mask, and they are hired to treat the cattle, 500 cattle per farm every day, and then the acaricide goes over here and goes to this small river, and where it goes, it's killing all the insects, because it's toxic for all the insects, arthropods here, and it goes to the river, and we don't know the ecological damage that is causing. And this is because they have to use acaricides to treat cattle. This is a distribution of the ticks in Mexico. The ticks that transmit babesiosis is from here, of the tropics, because these are this is a tick from the tropics on the subtropical area over here. Only very high areas that they don't have ticks, but then that means that approximately 66% of the national territory is infested by ticks that transmit babesiosis. 66% of the cattle in Mexico, which are in these areas, are susceptible to this disease. So, to avoid the dissemination of the disease, vaccines are the most reliable, cost-effective, and ecologically friendly control method. But there are no recombinant vaccines for bovine babesiosis in the world. Nobody has developed a vaccine against bovine babesiosis. Some countries, they use live vaccines, like attenuated live parasites, but they have to feed them every day with erythrocytes of cattle. Live vaccines are very expensive, are unstable and unsafe, and they may cause co-infection with other pathogens. Producing live vaccines is very complicated. There's only 3 countries in the world that have live vaccines, and they cannot export them because of the dissemination of pathogens. But we decided that we should try to develop a vaccine against babesiosis. How do you develop a vaccine against bovine babesiosis? You have to understand the parasite first. You do that by studying how it infects the erythrocyte. It infects the erythrocyte by adhering using receptors on the membrane, and then it turns so that it comes in contact with the erythrocyte. It opens the erythrocyte membrane, and then it invades the erythrocyte. How do we prevent those merozoites for infected erythrocytes? That is what we thought we should study. If once the parasite goes inside, it divides in 2 merozoites. These merozoites mature, they break the membrane of the erythrocyte, and then go and invade another erythrocyte. And this happens in 2 to 3 hours, more or less. So in 10 days, the cattle basically doesn't have any erythrocytes because the parasite multiplies so fast. What we did is we studied the invasion cycle and identified molecules that the parasite used to attach. It used to reorient to the erythrocyte membrane. It used to penetrate the cell, the erythrocyte, and then it uses proteins once it is inside of the cell, and also when it goes to the tick, there are proteins that the parasite uses to reproduce sexually. So we identified all these proteins by using bioinformatics and a lot of amplification of genes in the genomes. And then we have a special strategy in our laboratory that consists of the selection, amplification, and cloning of genes of importance for Babesia. So what we do is we go to the field. It could be Chiapas or Veracruz. Last week we were to the Sierra in Calpan, in Querétaro. We collect lots of cattle that has ticks, and then we make sure that the cattle is infected with the parasite. We then go to the laboratory and extract the DNA of this parasite, and then by PCR, we amplify the sequence of the gene that codifies for the protein that the parasite uses to infect the erythrocytes. Once we amplify the gene, we purify the sequence of the DNA, and then we identify this epitope of this protein, right? So we have the multi-epitope protein here. These are the epitopes, but we use the coding sequences of these epitopes, and this coding sequence of this chimera gene, we insert it in a plasmid, in a bacterial plasmid, and then we get the plasmids with the inserted genes, and then we insert we transform the bacteria with the plasmids over here. So we have a colony of bacteria that have plasmids that have the gene inserted in there. This is the first step. And then what we do is we have this bacteria, grow them in a bioreactor here. Can you play the video? This is a bioreactor that we have in our laboratory. This bioreactor has the bacteria and is connected to a computer that maintains the temperature and the oxygen for the bacteria to grow and express this chimera protein. And then we purify, express and purify the recombinant proteins. How? We take the bacteria that is expressing the recombinant protein, lyse the bacteria, and then by affinity chromatography, we purify the chimera protein here, wash all the lyses of the bacteria, and we then collect the chimera protein. And this chimera protein, we emulsify it with adjuvant. Once we have the vaccine prepared already, then the idea is that we immunize cattle. The cattle should generate an immune response so that when the cattle is moved to the grass and the ticks infected and transmit the Babesia won't be able to invade the erythrocytes. So for that, we built in our facilities something that we call NIMFA in Spanish. NIMF, which stands for Nave de Infectómica Animal. So this NIMFA is a facility where we can contain animals here, isolated, completely isolated. It has a capacity for up to 20 cattle in individual pens. And the unit is maintained with a temperature and humidity, especially for tick development. So we can test tick vaccines or vaccines against Babesia. And we also have some pens where we can vaccinate the animals and then we move them to the NIMFA when we want to apply ticks on them. And we have performed trials, vaccine trials for several institutions, internationally, like University of Castilla-La Mancha in Spain, for Merck, now, before it was Interbred in the US, and then for the Biology Center in the Czech Republic. They have tested their vaccines in our facilities because our facilities comply first with bioethics protocols that are approved before we start each project, and the building follow the guidelines of the European Convention of the Protection of Vertebrate Animal Use, which means that it can have experimental animals in the unit with approved in any part of the world. So this is the chimera. This is the chimera protein that we developed in our laboratory. These are the antigens that I told you, and these are the epitopes of those antigens over here. And we developed the recombinant protein. We show that the bacteria express the protein. We purify the protein by affinity chromatography, and then we inoculate the protein to cattle. The cattle develop antibodies. We show that the cattle develop specific antibodies because the antibodies recognize the parasites inside of the erythrocytes. And then we tested the vaccine. We took 2 groups of cattle, one group with 5 cattle. It was immunized 3 times with the recombinant vaccine, and a group of 5 cattle was immunized only with adjuvant, no vaccine. 10 days after the last immunization, all the 10 cattle were challenged with 1 to the 8 intererythrocytic parasites of a virulent Babesia bigemina strain. Or were challenged with Babesia. And then we monitor the clinical symptoms like rectal temperature, packed cell volume, and the number of parasites in the blood. And then also we measure cytokines and some other parameters. What we observed in the production of antibodies was first that the control cattle, they never show any antibodies against the protein, but the immunized cattle in red, they generated antibodies after the first immunization, and the level of antibodies continued to increase until the day of challenge. For the rectal temperature is a measure to determine whether there is fever or no in cattle. If there is fever, that means that the cattle is sick. You determine that the cattle is sick because it has had 3 days of consecutive fever. And fever is defined in cattle to have 39.5 centigrades of rectal temperature, which means that all the counts over this line are considered fever if they are presented in 3 consecutive days. As you can see here, the control animals, most of them have fever for 3 days, some very high, and the immunized animals, none of them have fever for 3 or more days. Some of them have fever, but then they control the fever easily. So they did not require any treatment at all. When we measure the parasitemia is obtained by taking a sample of blood, making a smear, staining with Hiemsa, and counting the number of erythrocytes that are infected. So we count 2,000 erythrocytes per sample. So what we show here is, sorry, this is one. This is the infected erythrocytes here. All the control animals we started to see parasites from day 1 or 2 after the challenge, and then some of them started to rise, increase the number of infected erythrocytes. And then over here, some of them had a high parasitemia. So on day 9, they have to be treated because the life was in risk. So to avoid death, we treat, and we consider this experimental death in all of them. For the immunized cattle, they didn't show parasites up to several days after challenge. Some of them show parasites. Actually, all of them show parasites, but the counts of the parasites were very low, and they controlled the parasitemia themselves, and they did not require any challenge. When we measured the amount of erythrocytes in the blood, we saw that most of them started to lose erythrocytes over here, but on day 6, the control animals lost more red blood cells than the vaccinated animals. So that means that we actually could make these animals not get infected with not get disease. They got infected, but they did not get disease. So the conclusions of this work, I will tell you in a second, but then with that information, we could patent the sequence. If you want to patent the sequence, like a vaccine, you have to prove that the vaccine works. So the INPI asked us for the results of the challenge for the couple. So we had to demonstrate that the vaccine protected couple so that they could proceed with the registry. And this was the work of Diego Hernandez. Diego did a PhD in my laboratory. He developed the vaccine. He assembled all the sequence, analyzed all the peptides, the epitopes, and then we challenged the animals. He presented these results at the second Babesiosis meeting at Yale University some years ago. He got the first place for oral presentation for his research. You may not know, but Babesiosis was discovered in 1888. And since then, nobody had developed a recombinant vaccine against this disease. So that was pretty amazing. So we developed a multiple epitopic vaccine against bovine babesiosis that prevents disease. We believe we should improve the quality of the disease of the vaccine so that it prevents the animals from getting infected with the parasites. Chimera vaccines can be developed and produced by recombinant methodologies, but we need better tools to identify vaccine candidate antigens against ticks and pathogens transmitted by ticks. We need more and better vaccine formulations, but we also need more young people doing science and developing vaccines. We have in Mexico a very small amount of scientists working on vaccine development. And we saw when there was this COVID pandemic that there were very few laboratories that developed a vaccine. We do develop a vaccine. We did develop a vaccine against COVID in my laboratory. We patented also, but the funds were, the government didn't give us any funds. The university got some small funds, and we could not continue to the clinical trials. So with that, this is my group, the Vaccines and Immunology Research Laboratory, and I appreciate for your time. And thank you very much.
Are there any questions?
Okay.
Well, I got a question. While you were doing your experimentation, did you see any resistance to the vaccine in the cattle? Or from the beginning, it was so effective that you didn't see any resistance to the product?
You mean when we apply the vaccine? No, we did see some effects on the cattle. When we mix the adjuvant with the protein, you see that the cattle gets like an inflammation. So that is kind of a problem, but the cattle they don't suffer any pain at all. You know, the only problem is that us as persons, we don't like to see or to have a huge inflammation in any part of the body. But for cattle, they don't really have any problem. So we didn't see that we were worried about.
Another question is, where did you get the concept, or do you have another example of this chimera protein that it is having applied in other areas? Or it's just like a
No, the chimera vaccines have been used for many years now, but they don't do it the way we do it. It's like we have like this novel strategy because we test one epitope, we immunize animals, and we show that this epitope is inducing a protective immune response. And once we show that this works, then we use it to ensample the chimera. The other chimeras that are available, everybody produces it. It just takes a piece of a protein, get it together with another protein, and they don't test them individually. So we don't call it really a chimera because those parts of the proteins are not being tested before. So we decided that we should start testing epitope by epitope from every single antigen. The ones that are effective are used to assemble the chimera. The ones that are not effective are discarded. It takes a lot of time, like 4 years or 3 years.
I have a couple of questions. The first is, are there any side effects to the vaccine vaccines? Like, what is the immune response to the presence of all those different?
No, what we do first, especially when we try vaccines for humans or for animals that are very costly, like cattle, is that we test the vaccines first in mice. You have to prove that the mice, they don't have any adverse reaction before you try it in a cattle or in a human, especially. If the mice have a reaction, then you stop. Right. That's why making vaccines is very difficult because you have to make sure that first the mice, then the cattle, they don't have any adverse reaction. It's a lot of time and it's very expensive.
My other question was, like, how effective is the vaccine in comparison to other methods such as the use of parasites?
Yeah, this is a question that has been going for some time. You see, when you have a cattle that has 1,000 ticks on all the body, and you apply a parasite, 2 days later, the ticks are dead. They are no longer in the body. If you vaccinate, then you have to wait 10 days for this vaccine to be effective, and then another 10 days for the cattle to get antibodies. So by that time, the farmer gets very frustrated. He wants to get like rid of the problem like very fast. It's like a conceptual problem that they have. A vaccine is effective in the long term, and a parasite is effective right now. And they don't see it in the long term. That is like a problem that we have to convince them.
My final question is, do you have any ideas or any ways in which you can perhaps make the vaccine much more effective than what it is in its current state?
That's what we try to divide it to improve. What we try to do now, there is a disease in rabbits, viral hemorrhagic disease in rabbits. It's killing all the rabbits. It entered the US and it's spreading very easily on wild rabbits. It's killing all the rabbits. So we made a vaccine for the rabbits. But we said, we won't be able to inoculate the rabbits because they're wild. How do you wrap a rabbit? So we said, okay, let's give it in the food. How do you produce a vaccine in the food? Well, you make alfalfa recombinant, so that produces the vaccine. So when the rabbit is eating, it is immunizing itself. Something like that. It may be that you develop like a nanoparticle that covers the protein, and it makes that the rabbit doesn't destroy the protein in the gut. So you have to think of ways how to improve vaccines. Yeah,
So have you thought about work another type of animal or just with cats?
No. We started working with vaccines for rabies, and now we started working with vaccines against ticks, but from dogs. And last week we started working with ticks from horses. So we hope we can get some vaccines against those. But these are against the tick, not against the parasite.
And my question is that your research, did you get any like government foundation or support? Or it was your thing that?
We usually apply for funds in CONACYT, but it's very uncommon for us to get funded. So what we do is I have a collaboration with a Japanese university that give us some funds, and also for the USDA in the US. I have a collaborator that also we have a project with them. And recently I got a project from a collaborator in France. So in that way we can get money to buy all the cattle and the pigs and maintain all the pig colonies. It's kind of expensive.
So my question is more in an ethical way. You're making a vaccine and it's not ready yet, and you tried in an animal that has the disease. Would you consider it like animal torture?
You mean if I develop a vaccine and I immunize an animal without being the vaccine being registered?
Yeah, without being ready the vaccine.
Yeah, no, I'm not allowed to do that. My university doesn't allow me to do that. The only animals that I'm allowed to use are the animals that are approved by the ethical committee. And I have to specify the number of animals, the age, the gender, and everything. And what will I do with them after I finish the experiment? Which usually they go to slaughterhouse.
It's illegal to apply or distribute a vaccine that doesn't get the proper registration.
Yeah, it's illegal and it's very dangerous. And I would not. Yeah, because you know, if a vaccine has not been tested, how do you know that it's going to work on somebody or an animal? So if you try it, and if I immunize 10 cattle, like my experiment, then I need to get some funds to immunize 100 cattle. If my vaccine works, then I can register. But I cannot register my vaccine right now, and I cannot apply to a cattle in Veracruz because I will be in jail. Yeah, you can.
So is there any relation between the development of new vaccines and the parasitic resistance?
Yeah, it's exactly like COVID. COVID is mutated in a very fast rate. So the vaccines that were developed previously, they don't work against the strains that are circulating right now. So the companies have to make a vaccine against the new strains. It's like the influenza vaccine. You are vaccinated every day, every year, because there are new strains that the previous vaccine doesn't work against.
Oh, well, my name is Jennifer Velasco. I am on my fourth semester, and my question is, how long does it normally takes to develop and commercialize a vaccine for veterinary use, including the clinical trials and the permissions?
This vaccine, we started this vaccine in 2012. It's already 2024. The registry patent process has not been finished, and I cannot sell it yet. So it is usually around between 10 and 15 years.
Okay.
More or less. There and
that they don't have to recommend.
It is not common, it is not common, I don't know if I get your question, but let me know. It is not common in the market to have recombinant vaccines because in the veterinary field, it was easier to grow parasites or viruses, attenuate them, and make killed vaccines. And then you inoculate animals with those killed pathogens. That is kind of cheap. The problem with that is from the immunological point of view, those vaccines are not very good. And also, you only get a response against the immunodominant antigens of the killed parasites. Something that you can avoid using this chimera vaccine.
Hi, my name is Natalia, and my question is, you mentioned that you are already working on another vaccines for rabbits and horses, and how far along are you in?
Can you imagine, we went last week to collect ticks from a horse, and we found one tick. This tick already put, I think it put 200 eggs. Then we have to wait until the eggs hatch. Then when the larvae are mature, we have to put the larvae in a horse. So it transmits the disease. Then we have to get blood from the horse. So we can extract the DNA, amplify the genes, clone them, test them, and make the recombinant. So it's another 4 years until this stage. Yeah. Very slow. That's why I'm saying that we need more people to help us.
All right, well, first of all, thank you for such a great talk and for the knowledge that you're sharing with everyone here. I just want to highlight the fact, and I appreciate that you are telling our students that it takes a long time for developing this vaccine. And that the method refers to this, and then we do this, and then we need to wait for that. Because we live in a fast, immediate results culture, like with one click, we want to obtain what we want. Like buying something in Amazon, just click and then you have it. And I really appreciate that you are mentioning this to our students, because as students, we used to want that the results are immediate. Like, okay, just one line and I have it. And it's important to know that actually in the real life and in the real world, we need to wait and we need to work a lot. And it's not like that from one day to another. It's, as you mentioned, like 3 years of researching and trials and failing and like asking for funds and like being in relation with persons that are actually not in the field, because you need funds and you have to deal with different kind of persons, right?
Yeah, thank you very much. That's a good point. I have lately, most of the students I get in my lab, they approach me because they want to produce a vaccine. Everybody wants to have a vaccine because this patent that I show you, I'm sharing it with the area. If we sell it, it has to share it. But this vaccine started 10 years ago. So he got to make the last part. When somebody goes to my laboratory and says, I want to make a vaccine, okay, you will start it, but you won't finish. And then they don't like it. Because they, because they think that they want to have it, they want to be part of making the vaccine. It takes 10 years.
Yeah.
So
that's the real.
Yeah.
That's happened. Thank you very much.
Sure. Thanks.
So I'm guessing the vaccine is cattle industry, which is are there any further questions of these vaccinated cattle and then we consuming the meat? Are there any risks associated with that?
Yeah, that's a good point. No, the vaccine you have to test. It's very difficult for you to get a vaccine approved. You have to show that there is no residual of anything in any tissue. Actually, the vaccine that we try for rabbits, we have to take samples of every single organ of the rabbit, send it to pathology, and show that there is no histopathological damage of in any tissue, even the brain. You have to make sure that the vaccine is safe. It takes a lot of time, making the slides of the smears for all the routes on the brain. It's very complicated. You have to show that there is no unsafety in vaccination.
So in one of the graphs, we showed that there were some pretty big outliers in the graph, comparing the cattle that was the control and the cattle that you vaccinated. And the cattle that was vaccinated showed some pretty big outliers in a certain period of time in the development of the disease. Do you think there's some genetic reason behind that?
It could be, the cattle is very diverse. That is the problem with working with cattle. You cannot inbreed cattle like the mice. When you work with mice, all the mice are very similar genetically. So you expect a similar results in all of them. In cattle, no, because we buy cattle from the north part of Mexico, and they are very different. So maybe that genetically some are able to produce more antibodies and some are not. But you compare is the average of the production and compare it with the control. Yeah.
Well, my question is not that because I'm not studying anything about biology, but I have, so have you ever experienced an animal getting like, response to the vaccine different because of what you did in the animal? Like, I don't know, that the medicine didn't work or that type of incident inside the.
Yeah, we have many, it's not uncommon that when we work with rabbits, we immunize rabbits with different proteins, and there is one rabbit doesn't show any immune response at all, like zero. It's like if its immune system didn't see the antigen. But what we do is that we immunize the antigen in several rabbits. If all the other response, we assume that it is the rabbit that didn't respond. It's not the antigen. Well, thank you very much again.
Muy interesante.